Hello, everyone. I am Dr. Nervo. I am an assistant professor in the biology department. And today I'm going to talk to you a little bit about my research um, that I will be performing during the NSSURP 2021 program. So, a little bit about the research that I'll be conducting this summer. And so, in terms of a title project, my title tends to have this very long, lengthy, complicated uh, title, but the reality is my focus of my research is really thinking about how do organisms build an embryo? What do you need to be able to build an embryo? And really, how does cytoskeleton and adhesions help to organize tissue architecture? And so, how do organisms build diverse tissue? So, you start off as a fertilized egg, fusion of an egg cell and a sperm cell, which then results in a single cell. And so, we as mammals, for example, start off as a single cell organism that then through the process of embryogenesis undergoes all the specifications. So cells get specified to do certain jobs. Then those cells will start building within each other and divide and make more cells and then start creating tissues. And so tissues are made up of one or more cells or one or more cell types. And so you have all these different tissues within your body that result of all these divisions and the specification, right? And then these tissues result in having very different organizations and functions. And those functions are generally actually dictated by how those tissues look, right? What is their shape? And so, really, in biology, we often like to think about the fact that structure dictates function, right? And that's what I say in all my classes all the time. And so the same is true of tissues. And so your brain looks very different from your skeletal muscle, for example, right? And in order to get these different tissue types, cells must actually rearrange and reorganize as they're dividing in order to create these different tissue types. And so I am really interested in how these cellular rearrangements actually contribute to how tissues are formed. And one of these key components on how tissues are formed are cell adhesions, so how cells are connected to each other. And it's a really critical part of building an animal. And so cell tissues start off generally as really simple, really maintain regular shape and size. So the cells pretty much look the same, pretty simple organization. And the images here are part of a chick neurotube, which will become a spinal cord. And so cells within these tissues are not just simply glued together. They have these dynamic adhesions, and they're also changing shape while they're rearranging relative to their neighbors. And so you can end up going from this pretty simple organization very early in building an embryo, and then that tissue will become more complex during the later stages of building that embryo. So the later stages of embryogenesis. And so my goal is really to define the molecular machinery that's driving these different cellular rearrangements and organizations, these different cell shape changes, and how both those two things interact with each other to have cells communicate to be able to undergo this change from simple to complex. The cell type that I study this question in are epithelial cells. And so, what are epithelial cells? Epithelial cells are a polarized cell type, meaning that one side of the cell is very different from the other. Epithelial cells are tightly connected with each other through these adhesions represented with these little red circles here. And these adhesions are then connected to the cell cytoskeleton. The cytoskeleton is very similar to our actual skeleton, meaning that it helps the cells maintain shape or allows the ability of cells to move as well. And epithelial cells are one of the most prolific cell types in the body. And so therefore epithelial tissue is one of the most prolific cell tissue types in the body. And they have multiple jobs, right? They're pretty important for the functioning of most organisms. They line the air sacs of the lungs, allows you to breathe. They line the digestive tract, that allows for food absorption. So it allows you to absorb your food and get those nutrients in. And then one of the largest organs in the body, right, is made up of epithelial cells, so the skin as a protective barrier. So they're really important cell types that the structure of an epithelial layer, so those tightly adhered cells, are really important to how that tissue functions. And we know this through a lot of studies by a lot of people that if you disrupt how the epithelial layer is organized, meaning those cell shapes and then those connections, it can have detrimental effects. And so there's several human disorders and diseases that are caused by disruption of this epithelial cell shape and the ability of those epithelial cells to maintain tight connections. And this is just listing a few. So you really need to maintain this proper organization. So this is why I study this. And I study this epithelial cells and its ability to maintain a cell shape, rearrange relative to their neighbors, and maintain connection in Drosophila. And the reason that I can use Drosophila is that the a lot of the genes that are found in humans are also found in fruit flies, meaning that they're conserved. Right? And so there are great genetic tools as well in Drosophila. People have been using Drosophila to study genetics for years, and it's a streamlined model system. And so we actually share about 60% of our genome with Drosophila. So there's that 60% overlap. So we can use Drosophila as a really great model for human diseases and disorders or human function as well. And Drosophila has a really short lifespan, which makes it a great organism to use, especially for summer research. Right? Drosophila takes about 10 days to go from an embryo to an adult fly. And I stick to these stages of Drosophila life development, right? Embryogenesis and then early larval development. And so there are tons of amazing cellular events during embryonic development in Drosophila that we can study. This video that I'm going to show you here is a Drosophila embryo. Each one of these hexagonal little shapes are an epithelial cell. And what you're going to see is that the epithelial cells are going to change shape, change shapes, but also rearrange relative to their neighbors. And you're going to see this dynamic process, right? All the cells are starting to change shape, but they're maintaining connection with each other, right? Then this tissue undergoes a dramatic three-dimensional shape change. Right? So not only do you have the individual cells undergoing shape change, you have the tissue as a whole. Right? And so in order to change shape, and move, the cells must link their cytoskeleton to cell adhesions. So to make sure they don't go flying out of the tissue, right? They have to maintain some connection, but not too tight to where they can't move, right? So it's a delicate balance. This is another process in later embryogenesis where you have an epithelial sheet here, migrating together. So these are cohesive epithelial cells that are maintaining to create this barrier, this skin-like barrier that eventually is going to move and completely surround the embryo. And you can see it zippering, and now those two sheets have met each other and have closed, and now have encased the embryo, protecting everything that's inside the embryo from the outside. And so during this process of cell migration and cell rearrangements, there's actual tension and stress put on these cells. And we like to think about this idea of tension and stress when thinking about physics, but remember that biology is applied physics. So as a cell moving as a group, you have to maintain connection with your neighbor. These cells aren't floating in space, right? Just like those videos I showed you, there's a lot of dynamic processes happening at the same time. And so these cells are undergoing all types of stress. And the arrows here representing those stress or that tension that's put on the cells. And so you have to maintain connection with your neighbor. You also have to move and maintain connection with whatever it is you're traveling over if you're a cell, right? And maybe these are a different tissue type underneath the cell, right? And so you can think about this as a good example would be, or a metaphor would be playing tug of war. So each person on your team of your tug of war team represents an individual cell. So you have to maintain connection with the rope, but remember the rope is being pulled by the opposite team. And so there's that force pulling you in one direction while you're trying to pull in the opposite. You're also trying, like I said, trying to maintain contact with the rope, meaning maintain contact with your neighbors, right? But also maintain contact with the ground. You want your feet flying up under you. And so that's the same, these are the same forces that cells migrating as a group have to try to maintain. And so that brings me to the project options. So one of the project options is to determine if tension sensing proteins are available. So cells have adapted this amazing ability to deal with tension that I just spoke to you about by making and recruiting tension sensing proteins to areas of high stress. And so this project, you would be working on this process in early embryogenesis called germ band extension. This is the same video I showed you earlier. So germ band extends around. The second project would be discovering new proteins that may interact with known cytoskeleton adhesion intermediaries. And so we know quite a few players in this process, but we want to discover some new ones to see if there's any new proteins that we haven't figured out yet are part of this process. And this would be done in the process of late embryogenesis, or the process known as dorsal closure, which is this sheet migration that I showed you earlier. And so these are the two project options. I am okay with students working with each student working on their own project or working together. So with that, I will take any questions you have. If you email me at lnervo, this is cut off, I apologize. So even if you aren't sure if you're interested or not, you feel free to email me to ask more questions to see if you're interested. So no matter what level of question you have, I would be willing to take it. All right, thank you all for listening. I hope you have a great start to your spring semester, and I look forward to hearing from anyone interested. Thank you.